So thank you I'm Valerio from Prague, as you introduced me. Thank you. And thanks, John, for inviting us to this community and to this meeting. We are pretty new in the field. So we came to this community through another route. Let's say we originally we study about more like the evolution of the genetic code. But I want to bring you like through the speech how we get to modify Syn3A and to reduce the amino acid alphabet. So as I say, our lab is more specialized in the study of the evolution of genetic code. So how the genetic code evolved from the very beginning till like what we can consider the first cells, like the LUCA, the last common ancestral organism. And so the theory is that the genetic code is evolved from something really easy. So the original protein were composed of a really small easy set of amino acids. So we can imagine that the primordial protein or organism, they were not composed of the full set of amino acids, but just a small set of them. That we can assume that it's like this early amino acid that we can define early amino acid, and there are like only 10 of them. Between them, we can find the most easy one. Like glycine, alanine, leucine. It's most like probable they were more available in the primordial broad and were easily added to genetic code. Where the other one, they were more like expensive from the cost of energy, they were adding lately. Like for instance, the aromatic one, or like the sulfur containing like cysteine and methionine. So we can say that the ancient protein, most likely, they were composed of a really small set. And by the evolution, the genetic code acquired the more evolved, the more complex one, maybe also thanks to develop or some metabolism pathway that introduce and make them from the scratch. This evidence, it can be also find, like we can see some evidence in the modern cells. Like this is just an example of the E. coli proteome, where you can see the distribution of the different amino acids. And we can see that there is 10 set of amino acids. They are much more abundant in the modern proteome compared to the late set. But this what is interesting. It's not only that they are most abundant, but also the energy cost to make them was much less. So we can imagine if there was a primordial organism, it might be also evolved with not too many pathways. It would be much easier for them to produce like protein or like metabolism with this set rather than more evolved one. So the idea is that in the beginning, it was possible to have like protein with a small set of amino acids. And when the evolution required much more complicated pathway or function, they were acquired the new one. So, but to prove that, of course, it means that the protein has to evolve and the genetic code to be saved with this 10 amino acids. The protein composed of this one, they should have some function. Otherwise, they would not be selected and evolved. And so our first goal that we proved like in the past was to select two model protein, one RNA binding protein that is related to this like theoretical original lie where there was this RNA word. So there was so we have a like RNA binder and an enzyme. So we choose like we prove it. We try to prove that taking two modern protein, like an RNA binder and an enzyme, and if we substitute the late amino acid with the early one, so we kind of reverse evolve, so we make this modern protein in the 10E form, then we're still able to keep the function. And the first example was like using this RNA binding protein. So we select a protein from CLL11 from the ribosomal protein, the C domain, and through mRNA display, but I'm not talking about this technical detail, but just we create a library composed of only 10 amino acids. So we substitute all the late one with the early one. And after this mutation, this protein was still able to bind the native RNA. So we still keep the function. What was really interesting is this set of 10 amino acids of these 10 early, they were not positive one, no arginine, lysine. So it was interesting to find that the binding with RNA, that is like this negative charge, was provided by the magnesium. That the magnesium that also prebiotic available, it was like function as bridge. salt bridge between the aspartic residues and the phosphate of the protein. Then we decide, okay, we check if it's possible that the binder protein can still bind RNA, but okay, what about enzyme? That it's more like diffusing. And we did similar experiment. In this case, we remove all the aromatic residues from the decarboxylate, like from the phosphocoenzyme A kinase, and so we substitute all the aromatic amino acid with leucine, with that is part of the 10E amino acid. And even though the catalytic activity was reduced, the enzyme was still able to catalyze the reaction and bind the ATP. That was one of the important cofactors. So the first step of our path, our journey, was to establish that yes, it's possible that protein composed of a reduced set of amino acids, only 10 of them, can still preserve a function. That can be a binding, can be an enzyme. Of course, much worse than the original one, the wild type, but still able to perform it. Thanks to like the presence of cofactor or metal ions that assist. That it also makes sense in the logic of a prebiotic world where there was like cofactor or like ions that were pretty common in the media, let's say, in the environment. And now what brings us here is the next step. So once a protein can be still function with a reduced set of amino acids, can be an entire metabolic pathway, or can be a cells reduced to reduce its alphabet. So from 20, maybe not, of course, not extreme like only using this set of 10 amino acids, but reduce the set of amino acids from 19, 18, 70, to find the minimum set of amino acids that the cell can support. And to do that, so we decide to prove that is still possible, that we can have a functional cells with a reduced set of amino acids. We decide to use reduced cells. And here we are using like the Mycoplasma JCVI-Syn3B in particular in this case. Of course, I'm not here to introduce, I think everyone knows probably even more than us. So it's like, so we decide this because it's already a synthetic organism. The genome, it's already small compared to the other one. So it will be much easier from the technical point of view to reduce even more the eventual size, so to modify it. It's only 490 proteins, so technically it will be much easier. But first of all, we need to decide which amino acid to remove, because of course we cannot remove all of them. So the first step of our project, how we imagine our project, it will be like a basic bioinformatics tool. So we will use like different tool, can be Rosetta or other one that will be developed in our lab to estimate the energy of every single substitution inside a protein. So let's imagine that we have a protein, we decide to substitute the phenylalanine with other, I don't know, with tyrosine or with other substitute, and we calculate for each mutation the energy, and we have the overall energy of the single protein for, I don't know, for phenylalanine as an example. Then we repeat this process for all the proteome, in the case of Syn3, and we can calculate the general fitness of the cells. And we can do this for every single amino acid. And in the end, we can calculate the general fitness in the case of every single amino acid can be substituted. So in this way, we can select which candidate to use. For instance, can be phenylalanine, tryptophan. So we can select the first amino acid to remove from the cells, and still holding the general fitness. Of course, this will be just bioinformatics study, but it would help us to choose which amino acid to remove like one by one. It all will be like all together, but just one amino acid per time. And once we select the first target, can be phenylalanine, cysteine, tryptophan, then there will be like the technical part, the method. So one, it will be like the oligo recombineering. So thanks to different paper already published in Mycoplasma, for Mycoplasma pneumoniae, or for like the same C3, it's possible to recombine, to use this to engineerize this the genome through like recombinase GP35, this like recombinase of single strand oligo. So in this way, we can substitute like introduce mutation inside the genome. Of course, this part also need to optimize. Most of the method, like the most famous one, the sure one, is like optimized only for pneumonia. But that will be our part, like try to optimize the other methodology for other mycoplasma for the Syn3A. And in parallel, that it's more like, and this approach is more like focus, so we can introduce an oligo with the precise mutation that we can calculate through the bioinformatic part. And in parallel, there will be a laboratory evolution. So let's leave the cells to decide what is the best and push toward like maybe a substitution on the end. So we can go in parallel with different strain, with different of like Mycoplasma Syn3A. So in one, we can start with tryptophan, isoleucine, phenylalanine, and for each amino acid. And do this library evolution, continuous series evolution, we see if the microorganism is able to adapt to absence of the one amino acid and in which way. Of course, in our our goal is that it start to maybe this way, easiest way is to reduce the number of that amino acid in the genome, maybe some modification in tRNA synthetase. There are not so many, this is the good thing of Syn3A, there are not so many escaper way, where like in E. coli, there will be activated much more safer pathway to just for the production or of some amino acid, but in Syn3A, that it's minimal genome, probably there won't be many of them. So maybe we can push the organism to reduce by himself the content in percentage, the content of that amino acid in order to survive. And every serial, there will be like more than 100 generation, and we follow with like whole genome sequencing, amino acid analysis, and repeatedly for as much many generation. So this is our goal. So let's say it's still a project on paper. We still didn't have any data for it. It's just our goal, try to reduce Syn3A not in terms of genome, but also in terms of proteome. And I would like really thanks John Glass that provide us the strain, invite us in the community, and is giving us really great help in the beginning. And I want to thanks our group, my group, and in particular all the team that is working on this project, and all our collaboration all around the world. Thank you very much for having us here.
Valerio, this is terrific. Questions for Valerio?
Yeah, Valerio, Kim Wise again. Yeah, I had a question for you about the whole approach. Back in the 70s and 80s, Alan Rodwell worked out a defined medium for the parent organism that became Syn 1.0. That's mycoides, one of the mycoides strains. And in that work, he found that every amino acid was essential. There was no auxotrophy. And of course, the way he did the experiments may have been a little less elegant than what you're planning. But you might look back at those because, they simply didn't grow without each amino acid. And he did, I don't know what all the combinations were, but that was the conclusion of the paper. So you might just, it's Alan Rodwell, Rodwell. I could send you a couple of references, but it might be good just as background to see how that relates to what you're going to be doing.
Okay. For the moment, we are conducting this experiment also in parallel with E. coli. And of course, like the metabolism and the escaper, it's much easier to achieve. So, in E. coli is working. But of course, like Syn3, it's a new genome, it's much more simple. So for sure, you cannot activate this ecton pathway. And we need to try. But thanks, we can give a look to the material. And
yeah, it might be helpful.
Yeah, thank you.
Valerio, James Dobbins Speck in Birmingham, Alabama. Do you think there are any ancient amino acids that went extinct that didn't survive to this present day? Obviously 10 of them did in your thinking, but you think there was a bigger set than that 10 and some of them just didn't make it or evolved into the ones we see now?
Yes, I mean, what we can say now, it's like a kind of black box. So we can assume what was in the beginning. I didn't enter too much in detail, but yeah, there are different proof. There is like from asteroid or from sampling from ocean, deep ocean, microvents. And there are like this team, they were more abundant, but of course they were like much more like also these non-canonical amino acids. So what we know is what was the initial pool and the final one. So it's also totally possible that there were other ones that are disappearing because, I don't know, can imagine ornithine, for instance, can be there and by time can be substituted with arginine. So it's hard to predict the trajectory. But let's say these 10E, they were the most abundant in the prebiotic world. So most likely they were there. And sure, for sure, there will be other ones that probably they were there and the evolution just selected by time, but also by the evolution pressure. Like it depends also how the environment change and the requirement over the million of years, so billion of years. So yes, it's totally possible. And we still, in our organism, we can still find some of these non-canonical amino acids, like this residue, like you can mention ornithine, and some of them are really still common. So it's possible they were there and just they didn't make it. Oh, another thing on the previous question, just thinking about like of course we will not try to starve the organism from the beginning. So it would be like some gradual adaptation. So like starting from high concentration, for instance, of tryptophan or like phenylalanine or one of the amino acid, and by time slowly go down to give maybe time to the organism to adapt. So that one is more the plan line. But of course, like if this I would be happy to read the paper and maybe we can learn from it.
Valerio, could you, so I see the advantage of our minimal cell in that it in that it doesn't make any amino acids. So it allows you to control, if you make a defined media, you can control the concentration of all of the amino acids. Are there ways that we might engineer the cell to by adding, for instance, adding pathways that are to the minimal cell that are not essential for life, but essential for but that you could push towards evolving in a way that you wanted without like killing the cell, but the pathway could accelerate growth that you could select for it that way. And thinking about minimal cell metabolism, for instance, if we look at the flux balance models that have been generated by Zanluthi Schulten's group at the University of Illinois, there are places that we could remove a gene and make the cell grow substantially slower. Mm-hmm. And then you could add a gene back that the only way it's going to work is if it evolves in the way that you want it to. So for instance, the model predicts, and Ronald Rodriguez will talk about this in a little bit. The model predicts that without a transaldolase, the cell would divide in 8 hours, and with a transaldolase, the cell divides in 2 hours. Now, transaldolase is in the pentose phosphate pathway. There is nothing in our genome that looks like a transaldolase, and Ronald will talk about this. We've identified another protein that may not be a transaldolase, but is somehow maybe necessary for this more rapid growth. And so here's a thing that if you put a messed up version of this other protein in that had eliminated some of the amino acids that you wanted, but that the cell would have to be, I guess that the cell would have to find a way, maybe in the absence of, maybe in, okay, or you take away critical proteins, critical amino acids for that protein. I don't know. I need to think this through, but there might be a way that we could work together to build something that might accelerate your testing a little bit.
I mean, the idea, I mean, like what they can work on, or like maybe you can engineerize, or you can try to maybe cripple like the tRNA synthase, for instance, in somehow, because in this way, if you have like a pressure selection from outside that it's depleting of some amino acid, and on the other side, there is like some more or less specific tRNAs, so you can push toward the error. It can incorporate inside the other amino acid, like I can imagine, I don't know, a phenylalanine or a tryptophan, phenylalanine instead of tryptophan, or tyrosine instead of that, some more similar one, and you can push the cells to do that. And this is the good thing with the Syn3, because in E. coli, there will be always some, it's too much evolved and big, like metabolism, there will be always a second way to generate. But here, the cell cannot escape too much. It's true that on one side, it can grow, it can die, it cannot do nothing. But if it grows, I think there are not too many ways. I think one of that would be or reduce the number of the amino acid or die. So, but for sure, it need the help. So we can think about what some tRNA synthesis or some other pathway that can push the cells to improve this mutation.